Hi guys, my name is Nithmi Thisakya and this video will be on the final sections of Unit 9, the microbiology lesson, relevant to the national level curriculum. I hope this video will come in handy and assist you in the learning process. So in this video, we will study about the microbiology of domestic water and wastewater, then about solid waste treatment, and finally about microorganisms and food. First, let's see about the contamination routes of drinking water. Contaminated water means water unsuitable for consumption. So, water gets contaminated mainly by two methods, and they are first by infectious disease carrying organisms. The second method is by chemical pollutants. Water contamination by chemical pollutants occur due to industrial activities, agricultural activities, and domestic activities. Most of these chemical pollutants are non-biodegradable or resistant to biodegradation. So, some of these chemical pollutants can also leach from soil surface into underground water and contaminate underground water. Let's see an example for contamination of water by chemical pollutants. You know, the fertilizer that we use in agriculture has high amounts of nitrates and phosphates in them. So, due to excess use of these fertilizers, these nitrates and phosphates may get washed away with rain and accumulate in water bodies. This will result in an increase in concentration of these ions in water bodies, leading to an increase in nutrient amount. So this will result in the growth of microorganisms such as cyanobacteria and algae in these water bodies. This process or phenomenon is known as eutrophication. The dense growth of algae on these water bodies are known as algal blooms. So water from such water bodies are not suitable for consumption. This is because these water bodies may contain toxic water. Chemical pollutants released from industries also can cause water contamination. Now let's talk about using microorganisms as indicators of water quality. Water can contain pathogens such as Salmonella, Shigella, Vibrio. So it is essential to determine the presence of these microorganisms or pathogens in water. But determining these pathogens in water by counting these pathogens is not an effective method. The reason for this is First reason is because even in contaminated water, these pathogens are present in very small amounts. So we may not find them in our test sample. The second reason is that the laboratory tests used to determine these pathogens separately is a time-consuming process. So it will be very late to prevent the outbreak of the disease. So we use an alternative method instead. Instead of pathogenic microorganisms, we detect the presence of indicator organisms in water. These indicator organisms are present in large amounts in fecal matter. So if we find indicator organisms in a water sample, it means that the water sample is contaminated with fecal matter. So the presence of indicator organisms in a water sample ensures the contamination of water. In countries like Sri Lanka, we use coliform bacteria as the indicator organism. Coliform bacteria are a type of bacteria present in the colon. So these coliform bacteria are present in large amounts in fecal matter. Coliform bacteria can also be present in soil and plants. But there are methods to determine those present in fecal matter from those present in plants and soil. So it won't be an issue. The test used to determine the presence of coliform bacteria in a water sample is known as the coliform test. Routine coliform tests are carried out by the laboratories of National Water Supply and Drainage Board. Now let's talk about waterborne diseases. Waterborne diseases are diseases occurring as a result of consumption of contaminated water. A main reason for waterborne diseases are the pathogens present in contaminated water. Examples for waterborne diseases are typhoid, paratyphoid, cholera, dysentery, gastroenteritis. Consumption of contaminated water containing pathogenic bacteria or microorganisms can lead to infections in the intestinal tract. Now we'll talk about the process of drinking water treatment. Water from water bodies is not suitable to use as drinking water directly. This is because it may contain pathogens. So first, water must undergo a purification process before we use water for drinking. This purification process is known as drinking water treatment. Water purification does not necessarily mean sterilization of water. During water purification, the harmful or the pathogenic microorganisms are removed. So the process of drinking water treatment consists of three main steps, and they are sedimentation and coagulation, filtration, and finally disinfection. The first step of drinking water treatment is sedimentation and coagulation. During this process, first water is filled into holding tanks and they are stored in the holding tanks. In this step, the large particles that are present in water get settled at the bottom. So this sedimentation is enhanced by addition of alum. Alum means aluminium, potassium sulfate. So this alum is a sticky precipitate. With alum, most of the microorganisms and the finely suspended matter get settled at the bottom. The second step of drinking water treatment process is filtration. During filtration, the water flowing out from sedimentation and coagulation process is sent through beds of sand. And when water is sent through beds of sand, it gets filtered and the microorganisms that are present in water gets trapped by these soil particles to the surface of these soil particles. Filtration process removes most of the protozoans, cysts and other microorganisms. By this method, we can remove about 99% of bacteria. The filtration process removes 99% of bacteria present in water. In new urban water treatment plants, active carbon is also used. When active carbon is used, we can remove toxic chemicals in water. The final step of the process of drinking water treatment is disinfection. Here, disinfection can be done mainly by two methods, and they are by chlorination, and the second method is disinfection using ozone. Chlorination destroys the microorganisms present in water. In disinfection using ozone, because ozone is highly reactive, the disinfection process occurs by oxidation. Compared to chlorination, disinfection using ozone is more preferable because ozone is colorless and odorless. So now let's talk about the principles and the main steps of industrial wastewater purification. Now let's talk about wastewater management. Wastewater means used water from domestic, agricultural and industrial activities. So water from toilets, industries, washing are considered as wastewater. Wastewater from industries are not directly allowed to flow into natural water bodies without purification, or else it will contaminate the water bodies. So first, the industrial wastewater is purified. This process of industrial wastewater purification involves two main steps, and they are the primary treatment and the secondary treatment. The first step of industrial wastewater purification is the primary treatment. During primary treatment, stepwise purification of water takes place. First, the large floating materials present in the water are screened out. Then, sand is removed. Then the oil and grease present in water is removed. Then, solid matter is allowed to settle in sedimentation tanks. The settled solid matter or the sludge is then collected and removed. You must know that in primary treatment, no biological activity is used. 25 to 35% of organic matter present in water is removed during the primary treatment. The second step of industrial wastewater purification is the secondary treatment. Liquid from primary treatment enters secondary treatment. During secondary treatment, aeration takes place. Aeration means mixing of water with air. So this facilitates the growth of aerobic bacteria. This results in rapid microbial oxidation. The two main methods used for aeration are activated sludge method and the trickling filter method. During this step, 75 to 95% of organic matter present in water is oxidized. After aeration, disinfection of water takes place. The disinfected water is then sent into natural waters. The remaining sludge is then sent to the anaerobic sludge digester, where anaerobic digestion of sludge takes place. Fertilizer and biogas are produced as byproducts during this anaerobic decomposition process. Let's move on to the next section, solid waste treatment. In this section, we'll learn first about the nature of solid waste, then about the environmental and hygienic importance of recycling solid waste, and finally about solid waste management. Now let's talk about the nature of solid wastes. Solid wastes can be of plant and animal origin or of synthetic origin. Plant and animal residues, kitchen waste, garden wastes are examples of solid wastes having plant and animal origin. These solid wastes are organic, so they are biodegradable. Plastic, polythene, glass are examples of solid wastes having synthetic origin. Poor management of solid waste can affect community health and environmental safety. It can also lead to soil, water, and air pollution. Now let's talk about the environmental and hygienic importance of recycling solid wastes. As I mentioned earlier, poor solid waste management affects community health and environmental safety. Now let's see some examples for poor solid waste management and how it has affected community health and environmental safety. Open dumping of solid waste creates breeding ground for insects such as flies, mosquitoes, and animals such as rats. These organisms can act as vectors for various infectious diseases such as chikungunya, foodborne diseases, and leptospirosis. Poor solid waste management can also cause contamination of water. Disposing of solid waste into water bodies can result in the spread of waterborne diseases such as typhoid, cholera, dysentery, and gastroenteritis. Also, piling of solid waste results in anaerobic decomposition of this solid waste. This will result in production of gases such as methane, and this can lead to explosions and fire. Another effect of poor solid waste management is groundwater pollution. Various liquid chemicals produced during the chemical and biological processes taking place during decomposition of solid waste can leach through the soil layers and contaminate groundwater. Now let's talk about solid waste management. In this lesson, we will learn about three main methods of solid waste management. They are separation and recycling, degradation, and sanitary landfilling. In sorting and recycling, what we do is we sort solid waste according to their nature into separate containers as recyclable waste and non-recyclable waste. Degradation is the second method of solid waste management. In degradation, readily degradable solid waste such as kitchen waste and garden waste is subjected to natural microbial degradation by a process known as composting. So the compost produced can be used for agriculture and gardening activities. The anaerobic conditions in composting can facilitate the growth of methanogen bacteria. So the methane produced can also be used to produce electricity. So this can be used as a method of additional income. Sanitary landfilling is the most popular form and the least expensive method of solid waste management. So nearly 4/5 of municipal waste is disposed in this manner. In sanitary landfilling, solid waste is spread as layers on an open land and then it is covered by soil. So the degradation occurs mainly by biological and chemical processes, and this will result in formation of solid, liquid and gaseous products. Sanitary landfilling is not suitable in areas with high groundwater level. Now we'll move on to the final section, microorganisms and food. In this section, we'll learn why food is spoiled by microorganisms. Then we'll study about the physical, chemical and biological changes that occur in food spoilage. Then we'll learn about the external and internal factors that affect food spoilage. Then we'll learn about the foodborne pathogens of humans. And finally, we'll learn about the effects of food spoilage on human health. So why is food spoiled by microorganisms? The food we use for human consumption is always of plant and animal origin. Microorganisms inhabit every niche on earth. Plants and animals are no exception. So, the food contains a natural microbiota. Also, the food can get contaminated with microorganisms during handling and processing. So these natural microbiota and the microorganisms that enter food during contamination can cause food spoilage. This is because the nutrients present in food are the same nutrients required for the growth of microorganisms. So food acts as a nutrient medium, a culture medium for microbial growth, such as nutrient broth. Bacteria, molds and yeasts can grow in this broth. Now let's talk about the physical, chemical and biological changes that occur during food spoilage. Growth of microorganisms in food and changing of chemical and biological structures of these food due to the microbial activity making food unsuitable for consumption is known as food spoilage. Food spoilage mainly occurs by heterotrophic bacteria and fungi. So these heterotrophic bacteria and fungi grow on food and changes the nutrients in food like carbohydrates, proteins and lipids using various extracellular enzymes such as amylase, pectinase, cellulase, lipase, protease. So this activity of microorganisms on food causing physical, chemical and biological changes results in food spoilage. Some of the major chemical changes occurring during food spoilage are putrefaction, fermentation and rancidity. These chemical changes occur as a result of the activity of extracellular enzymes secreted by microorganisms onto food. So during putrefaction, the proteolytic enzymes convert proteins into amino acids, amines, ammonia and hydrogen sulfide. During fermentation, amylase converts complex carbohydrates in food into simple carbohydrates. Then these simple carbohydrates are converted into carbohydrate food acids, alcohols and gases by enzymes secreted by saccharolytic microorganisms. In rancidity, lipids are converted into fatty acids and glycerol by lipolytic microorganisms. Some of the major physical changes taking place during food spoilage are softening of food, pigmentation, ruppiness, slime or gum formation, and toxin accumulation. Pigmentation means the changing of the color of food. Ruppiness means the development of a sticky texture. So now let's learn about the external factors that affect food spoilage. Some of the external factors that affect food spoilage are temperature of storage, relative humidity, the presence and concentrations of gases in air. Now let's see how temperature of storage affects food spoilage. Temperature affects the growth of microorganisms. In low temperatures, the growth of microorganisms is slow, so the spoilage process is slow. In ambient temperatures, the growth of microorganisms is high, so the spoilage occurs easily at ambient temperatures. Food spoilage can also occur at very low temperatures by psychrophilic bacteria. Relative humidity of the environment also affects food spoilage. Relative humidity means the availability of moisture in the environment. Since moisture also affects microbial growth, high moisture containing food are subjected to spoilage easily. So, food with low moisture content should not be placed in environments having high relative humidity because the food can pick up moisture easily, so it can get spoiled easily. The presence and concentration of gases like oxygen in air also affects food spoilage. Both aerobic and non-aerobic microorganisms can cause food spoilage. Now let's see some of the internal factors that affect food spoilage. So some of the internal factors that affect food spoilage are pH, the moisture content of food, the nutrient content of food, the biological structure of food. The pH in food affects microbial spoilage. Most microorganisms grow around the pH value of 7. Only a very few microorganisms grow below the pH value of 4. Molds and yeasts have a wide range of pH ranging from 2 to 7. Bacteria in general grow around pH values 5 and 7. Acidic fruits, which have low pH such as lime, orange, banana, are spoiled by molds and yeasts. Food like beef, chicken, fish and milk are spoiled by bacteria, molds and yeasts. The moisture content in food also affects food spoilage. Moisture is essential for the growth of microorganisms. So high moisture containing food such as meat and fish are easily spoiled by bacteria. Low moisture containing food such as biscuits and bread are spoiled by molds. Drying is one of the oldest methods of food preservation, and this is based on decreasing the moisture content in food. Dry food such as dry milk are not easily spoiled because of its reduced moisture content. Salty and sugary food are also not easily spoiled because of their reduced moisture content. Salty food is spoiled by halophilic bacteria. Sugary food is spoiled by osmophilic and xerophilic molds and yeasts. The nutrient content in food also affects food spoilage. Water, vitamins, minerals, and nitrogen is essential for the growth of microorganisms. So, in general, nutrient-rich food such as milk and meat are easily spoiled by microorganisms. The biological structure of food also affects microbial spoilage. The natural covering of some food, such as the outer covering of fruits and the shell of egg, prevents food spoilage by preventing the entry and damage caused by spoilage microorganisms. Let us see some of the foodborne diseases and their disease-causing pathogens. Here you can see a few examples for foodborne diseases and the pathogens responsible for those diseases. Now let's learn about the adverse effects of food spoilage on human health. During food spoilage, microorganisms grow and multiply on food. So these microorganisms release toxic substances onto food. When food containing these toxic substances or the food containing these microorganisms is consumed, it will affect human health. So this will lead to foodborne infections, food intoxication, foodborne illnesses. During foodborne infections, the pathogen enters the body due to consumption of spoiled food. So the pathogen enters the body of the host and grows inside the host, producing toxic chemicals, toxic substances. So examples for some of the foodborne infections are typhoid, cholera, dysentery. What happens during food intoxication is the food already containing toxins is consumed. So the symptoms of food intoxication can be observed within a short time. Examples for food intoxication are food poisoning, botulism, aflatoxin. Viruses such as enteroviruses can also cause foodborne illnesses.